Hey, about 2 cents, Mr. Yance. I'm going to show you today and how we load a DNA specimen into what we call a electrophoresis gel. It sounds very simple. It's not as simple as it might sound. So I'll go through a little bit on the different parts that we'll be doing, and we're going to do a practice run today. I always like to do this so that when we do the real thing, we don't have as many miscues. We want to try to avoid that if we can, because I only got so much of these DNA specimens, and we don't want to lose them. Okay, so what we have, if you look very closely at this vial, this is what we'll be using tomorrow, or not Wednesday and Thursday. You'll see there's a dark blue little triangle. We tap it on the table and we get to the bottom, and that's what we're going to extract out of this tube. And then what we're going to do is we're going to use this thing called a pipette, which what we'll do tomorrow and today is you put a disposable tip on there. So every time you load a gel, you need to put a new one on. Today, we're just going to use one because I don't want to waste them. But tomorrow, every person or everybody that loads a specimen will use a different tip. That way we don't cross-contaminate, if it makes sense. And then there's a little button on these pipettes that if you can see it right here, this button right here, you just push it down and boop, you eject and throw away your disposable tip. Okay. The tricky part with gel electrophoresis is if you look in this little dish, I have what I call it's got a so many mils of something called a buffer, electrophoresis buffer, which is kind of a saltwater solution, which helps conduct current because we're going to run high voltage electricity through this gel, and it kind of helps with that. The problem is it sits above this gel, and the gel's really fragile. It's made out of sugar, so it's like Jello. Okay, now the ones we're using today are thicker than that, but tomorrow it'll be a gel that looks like a bowl of Jello. All right, and you'll see these little wells. If you look down in there, you'll see little tiny holes. Okay, and in these holes, hang on a second. Let me go get I'll pause this for just a second. So this is what the gel looks like. It'll be pretty close to this thickness, maybe about that thick. And then you'll notice it's got little holes in it. And what we want to do is we want to get the DNA into these wells. Now here's the tricky part. I don't want to poke down through these and poke it out the other side, or it's going to leak into my buffer and I'm going to lose it. And the thing that most students do is when they depress this big button up here, which releases the gel, you want to get it in the well, not too high either, because that'll disperse into the fluid. So it's tricky. So what I want you to do today is all of you load some gels. This is just a practice stuff. I have some dye that doesn't have any DNA in it. I have plenty of it. You can see it here in this vial. We're going to extract it out and load it, and we're going to see who does it the best. So if you got somebody in your group that's really good at it, you might have them load all the gels. If there's all of you are good at it, you can take turns. I don't care. But the biggest thing is 2 groups will go Wednesday and 2 groups will go Thursday because I only have 2 machines, electrophoresis machines. I just want to make sure that we don't mess up and have miscues and lose our samples, because there's only so much of this DNA. This DNA, you can see it in this tube. I don't want to lose it if I can help it. All right. So what you're going to do is you're going to take this. You're going to put your tip on. Okay. You're going to have gloves on because this stuff stains and it's not harmful. But you have a big button here that you're going to go into your vial. You're going to depress it, and you're going to suck that stuff up. And you can see how I've sucked it up into the end of the tube. Okay. The technique that most of the time we use, and this is good stuff to learn for any lab that we do in chemistry or biology, is I like to use my offhand to steady things because people tend to shake. And you're going to go down into that well, and you're going to get it where you think it is, and you're going to depress and gently lift that thing out. And if you look really close down in there, you actually can see where I've loaded that thing. Okay. So one more time, you're going to go in, different tip if you're really doing this. You're going to extract, go in, get it into the well, and depress and lift it out. Okay. If you see a bunch of it leak out into the fluid, then you might want to try it again. Okay. Um, it's a lot harder than it might look, but the biggest thing is keep this hand kind of as a steady guide and go down in, and you'll see and depress and then release. And you got to be careful because you got to depress and keep it down, because if you depress and let up, you suck it back up into the tube. Okay, so practice it. That's what we're going to do today. We're going to have plenty of time. That's why I always like to do this lab. Practice it. When I started practicing, it went much better when we did the real thing. These are really nice pipettes. They're pre-programmed. They put in the right amount. I got that all set for you. All of you have one of these, so I'll break you up into 4 groups today. Wear your mask, put gloves on, and we're going to see if we can load these gels. And we'll see who's good at it. Kind of practice it. Again, it's a little more tricky than you might think, but I think we'll do just fine. Okay? Hey, have a great day. We'll talk to you later. See ya. Bye.